Imagine standing on the surface of a glacier. All around you, ice stretches toward the horizon in shades of white and pale blue, carved by wind into ripples and ridges. The air is thin and cold. The silence is absolute. At first glance, you might think you're standing on a desert of frozen water, a place where nothing could possibly survive. But beneath your boots, within that ice, and even floating in the thin films of meltwater between ice crystals, billions upon billions of living things are going about their lives. Some are so small you'd need a microscope to see them. Others are just visible to the naked eye. Tiny dark threads wriggling across the frozen surface at dusk. Together, they make up one of Earth's most extreme and least understood ecosystems. Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. If you like these videos, please consider subscribing to the channel and commenting below if you have any feedback or suggestions for topics of future episodes. Tonight, we're going inside glaciers. We're going to discover how life not only survives, but actually thrives in conditions that would kill most organisms within minutes. We'll meet bacteria that produce their own antifreeze, algae that paint snow the color of watermelon, and tiny worms that spend their entire lives at temperatures just above freezing. We'll descend through layers of ice to hidden lakes that have been sealed off from the world for thousands of years, where strange communities feed not on sunlight, but on the minerals dissolved in water heated by the earth below. And we'll learn why scientists are racing against time to document these microbial worlds before they vanish forever as glaciers melt across the planet. The story of life inside glaciers is a story about resilience. It's about organisms that have evolved remarkable strategies to cope with extreme cold, complete darkness, crushing pressure, and starvation-level nutrients. But it's also a story about interconnection, about how, even in the most isolated places, life finds a way to form communities, to cycle nutrients, and to transform the landscape around it. So, let's begin this journey into the ice, starting at the surface and working our way down into the frozen depths. For a long time, scientists thought of glaciers as essentially sterile. Ice was ice, frozen water with little to offer living organisms. But starting in the late 20th century, researchers began to realize that glaciers are actually teeming with life. In fact, it's now estimated that glaciers and ice sheets around the world contain as many as 10 to the 29th power cells. That's a 1 followed by 29 zeros. To put that in perspective, the total number of microbial cells in glacier ice is comparable to the entire biomass of all the soil in all the rainforests on Earth. Glaciers, it turns out, are not frozen wastelands. They are biomes in their own right, supporting diverse communities of microorganisms that have adapted to some of the harshest conditions imaginable. What makes a glacier habitable? At first, the question seems absurd. Glaciers are cold, often far below the freezing point of water. They're dark for much of the year, especially in polar regions, where winter brings months of continuous night. They're nutrient poor, with almost none of the organic matter or minerals that most life depends on. And yet, life persists. The key is that glaciers aren't just solid ice. They're dynamic, multi-layered environments with pockets of liquid water, thin films of moisture between ice crystals, and surfaces that melt and refreeze with the changing seasons. These microenvironments, scattered throughout the glacier like tiny oases in a frozen desert, provide just enough liquid water for microbial activity. And where there's liquid water, even in microscopic amounts, there can be life. Glaciers exist in several distinct layers, each with its own characteristics and its own microbial communities. At the very top is the snow layer, where fresh snow accumulates each winter. Below that is the firn, a transitional layer of packed snow that's slowly being compressed into ice. Deeper still is the glacier ice itself, solid and dense, moving slowly downhill under its own weight. And at the very bottom, where the ice meets the bedrock, there's a layer of meltwater and sediment, kept liquid by the pressure of the ice above and the heat from the earth below. Each of these layers supports different kinds of life, adapted to the specific conditions found there. The organisms that live in glaciers are known as psychrophiles, meaning they love the cold. Some are psychrotolerant, able to survive cold temperatures, but preferring warmer conditions. Others are true psychrophiles, thriving at temperatures near or below 0 degrees Celsius. These organisms have evolved a remarkable array of adaptations to cope with the cold. Their cell membranes are more fluid than those of organisms living in warmer environments, preventing them from becoming rigid and brittle in the cold. They produce special proteins called cold shock proteins and cold acclimation proteins that help their cellular machinery function at low temperatures. And perhaps most remarkably, many of them produce antifreeze proteins, molecules that bind to ice crystals and prevent them from growing large enough to damage cells. The study of glacier ecosystems has exploded in recent decades, driven partly by concerns about climate change and partly by the realization that these ecosystems might serve as models for life on other icy worlds. Moons like Europa, orbiting Jupiter, and Enceladus, orbiting Saturn, are covered in ice and may harbour liquid water beneath their frozen surfaces. If we want to understand what kinds of life might exist in those alien oceans, studying the microbes that live in Earth's glaciers is a good place to start. But there's another reason scientists are urgently studying glacier ecosystems now. Glaciers are melting all around the world, from the Alps to the Andes to the ice sheets of Greenland and Antarctica. Glaciers are retreating faster than at any time in recorded history. As they melt, the unique microbial communities that live within them are disappearing, many before they've even been fully documented. It's a race against time to catalogue this hidden biodiversity before it's gone. One of the most fascinating and well-studied microhabitats on glaciers is the cryoconite hole. The word comes from Greek, cryo meaning ice and conite meaning dust. Cryoconite holes form when dark particles, dust and soot and microorganisms land on the surface of a glacier. Because these particles are darker than the surrounding white ice, they absorb more solar radiation. This causes the ice beneath them to melt, and the particles sink down into the glacier, creating small cylindrical depressions filled with water. Over time, these depressions can grow to be several centimeters or even meters deep, though most are less than 10 centimeters across. What makes cryoconite holes so interesting is that they're not just holes in the ice, they're complete, self-contained ecosystems. At the bottom of each hole is a layer of sediment, the cryoconite itself, a mixture of mineral particles and organic matter. This sediment is dark, almost black, and it continues to absorb heat, keeping the water in the hole from freezing, even when the surrounding ice is is well below 0 degrees Celsius. The water in a cryoconite hole is remarkably clear, fed by meltwater from the surrounding ice, and it's teeming with life. The dominant organisms in most cryoconite holes are cyanobacteria, photosynthetic bacteria, that were once called blue-green algae. Cyanobacteria are ancient organisms, among the first to photosynthesize on Earth, and they're remarkably tough and adaptable. In cryoconite holes, they form mats and filaments at the bottom, using sunlight that filters down through the ice and water to produce energy. They're ecosystem engineers, shaping the environment around them in ways that benefit other organisms. The cyanobacteria produce sticky substances called extracellular polymeric substances, or EPS, that act like glue, binding together mineral particles, living microorganisms, and bits of organic matter into granules. These granules give cryoconite its characteristic texture and help stabilize the sediment at the bottom of the hole. But cyanobacteria aren't alone. Cryoconite holes also harbor diverse communities of heterotrophic bacteria, organisms that feed on organic matter rather than producing it through photosynthesis. These bacteria break down the dead cells of cyanobacteria and algae, recycling nutrients and making them available for new growth. There are also eukaryotic algae, particularly species from groups called chlamydomonadales and signematales. These algae are larger than bacteria and more complex, with distinct cell nuclei and other organelles. Some are green, some are golden, and some, as we'll discuss shortly, are brilliantly red. Perhaps most surprisingly, cryoconite holes are home to animals. Tiny animals, to be sure, but animals nonetheless. Rotifers, microscopic creatures with whirling crowns of cilia that they use to capture food, have been found in cryoconite holes in Antarctica. So have tardigrades, those nearly indestructible 8-legged microanimals, sometimes called water bears. And in a cryoconite hole on Ecology Glacier in Antarctica, scientists discovered an acari, a type of mite, marking the first time a glacier-dwelling mite had ever been found. These tiny invertebrates feed on bacteria and algae, forming the top of the food chain in these miniature worlds. The biological activity in cryoconite holes has real consequences for the glacier as a whole. Because the holes are darker than the surrounding ice, they absorb more heat, which causes more melting. The holes also act as reservoirs of nutrients and organic matter, which can be flushed out when the holes drain, carrying carbon and nitrogen downstream to lakes and rivers. In fact, cryoconite holes may contribute significantly to glacial wastage, the loss of ice mass over time, and to the biological colonization of ice-free areas as glaciers retreat. Scientists have even started thinking of cryoconite holes as natural laboratories, miniature ecosystems that can teach us about how microbial communities assemble, how they respond to environmental change, and how life persists in extreme conditions. One especially interesting finding about cryoconite holes is that they're remarkably similar all around the world. Whether you're on a glacier in the Arctic, in the Alps, in the Andes, or in Antarctica, the microbial communities in cryoconite holes have a lot in common. The same types of cyanobacteria dominate. The same general processes of photosynthesis, decomposition, and nutrient cycling occur. This consistency suggests that cryoconite holes are a fundamental feature of glacier ecosystems, shaped more by the physical properties of ice and light and temperature than by the specific history or geography of any particular glacier. But individual glaciers do have their own quirks. The exact mix of species in a cryoconite hole can vary depending on how isolated the glacier is, how much dust and organic matter blows in from surrounding areas, and what the local climate is like. Some cryoconite holes freeze shut in winter, sealing their microbial communities in stasis until the ice lid melts in spring. Others remain open year-round, allowing continuous exchange with the atmosphere and ongoing biological activity, even in the depths of winter. Scientists have also discovered that cryoconite holes can function like islands. In the same way that a real island in the ocean is an isolated patch of habitat surrounded by water, a cryoconite hole is an isolated patch of liquid water surrounded by solid ice. This isolation has consequences for the organisms living there. Bacteria and other microbes can't easily move from one cryoconite hole to another, so each hole develops its own slightly unique community. Over time, this isolation can lead to evolutionary divergence, with populations in different holes or on different glaciers becoming genetically distinct. It's a form of island biogeography, played out on a scale measured in centimeters rather than kilometers. If you were to visit a glacier in the high mountains during the summer, you might notice something strange. Patches of the snow, which should be pure white, are tinged pink or red, like a watermelon or a bowl of strawberries. This phenomenon is called watermelon snow, or red snow, or blood snow, and it's been known for thousands of years. Aristotle himself wrote about it. But for most of history, people didn't know what caused it. Some thought it was mineral deposits. Others thought it was pollen. It wasn't until the early 20th century that scientists finally agreed that the red color comes from algae, microscopic organisms that live in the snow and ice. The alga most responsible for watermelon snow is called Chlamydomonas nivalis, though recent studies have revealed that several related species, some now placed in a new genus called Sanguina, are also involved. Chlamydomonas nivalis is a green alga, closely related to the algae you might find in a pond or a puddle. But it has adapted to life in one of the coldest, most inhospitable environments on Earth. For most of the year, Chlamydomonas nivalis exists as a dormant cyst, a thick-walled resting cell buried in the snow. These cysts are not green, they're bright red, packed with a pigment called astaxanthin, the same compound that gives salmon and flamingos their pink color. Astaxanthin serves a crucial function for Chlamydomonas nivalis. At high altitudes and in polar regions, the intensity of ultraviolet radiation from the sun is extreme. Without protection, this radiation would damage the alga's DNA and its photosynthetic machinery. Astaxanthin acts like sunscreen, absorbing UV light and protecting the cell from harm. It also absorbs heat, which helps the algae stay active in cold conditions. The more astaxanthin a cell produces, the redder it becomes, and the more protected it is from the sun's harsh rays. In winter, when the algae are buried under meters of snow and starved of light, they remain dormant. But in spring, as the snow begins to melt and light reaches them, they wake up. The life cycle of Chlamydomonas nivalis is fascinating. When conditions are right, usually in early spring when meltwater becomes available and the days grow longer, the dormant red cysts germinate. They release smaller, green, motile cells that look much more like typical algae. These green cells have two flagella, whip-like appendages that they use to swim through the thin films of meltwater between snow grains. They make their way toward the surface of the snowpack, drawn upward by light. Once they reach the surface, they begin to photosynthesize, using sunlight to produce energy and grow. They divide, multiplying rapidly in the brief window of favorable conditions. And then, as summer progresses and the risk of freezing or desiccation increases, they transform back into red cysts, ready to endure another winter. The blooms of Chlamydomonas nivalis and related snow algae can be spectacular. In some places, entire fields of snow turn pink or red, visible from airplanes and even from satellites in space. A single millimeter of melted red snow can contain 500,000 algal cells. Multiply that across a snowfield kilometers wide, and you're talking about trillions upon trillions of organisms. These blooms have real consequences for the glaciers they grow on. Because the algae are darker than the snow, they lower the albedo, the reflectivity of the surface. A darker surface absorbs more solar radiation, which causes more melting. Studies have shown that the presence of snow algae can accelerate glacier melting by 5 to 15%, a significant amount that must be taken into account when modeling how quickly glaciers will disappear under climate change. Interestingly, watermelon snow is said to smell faintly like watermelon. Some sources even claim it tastes like watermelon, though eating it is not recommended. Astaxanthin, in addition to being a pigment and a UV protector, acts as a mild laxative. Early explorers and mountaineers who sampled the colourful snow reportedly regretted it. But beyond its curiosity value, watermelon snow is a reminder of just how pervasive life is on glaciers. Even the most barren-looking snowfield is home to millions of organisms, transforming the landscape in subtle but important ways. Snow algae are found all over the world, from the Sierra Nevada of California to the Alps of Europe to the glaciers of Antarctica. Different species dominate in different regions, reflecting local adaptations and evolutionary history. Some produce orange pigments instead of red. Others remain green, relying on other strategies to cope with UV radiation. But all of them share the challenge of living in a world that's frozen most of the time and scorched by sunlight when it's not. Their success is a testament to the power of evolution to find solutions even in the most unlikely places. To understand how life survives inside glaciers, we need to zoom in to the molecular level and ask a simple question. How do you stay alive when your entire world is freezing? For most organisms, the answer is you don't. Ice crystals that form inside cells puncture membranes, damage proteins, and kill the cell. But psychrophiles, the cold-loving organisms of glaciers, have evolved remarkable adaptations to prevent this from happening. Chief among these are antifreeze proteins, molecules that bind to ice crystals and stop them from growing. Antifreeze proteins were first discovered in fish living in the frigid waters of the Antarctic and Arctic oceans, where the water temperature can drop below the normal freezing point of fish blood. Scientists were puzzled. How could these fish swim around in sub-zero water without turning into fishy popsicles? The answer turned out to be antifreeze proteins in their blood. These proteins bind to the surface of tiny ice crystals, forming before they can grow large enough to cause damage. The ice crystals are still there, but they're small and harmless. The fish stays liquid. Since that initial discovery, antifreeze proteins have been found in a stunning variety of organisms, including insects, plants, fungi, and of course, the bacteria and algae that live in glaciers. Despite being discovered independently in different lineages, these proteins often share similar three-dimensional structures, suggesting that there are only a limited number of ways to build an effective ice-binding molecule. Some antifreeze proteins are shaped like helices, spiraling ribbons of amino acids. Others form flat sheets, beta helices that look like twisted ladders. Still others are more complex, with multiple domains and intricate folds. What all antifreeze proteins have in common is a flat surface studded with particular amino acids that can form hydrogen bonds with the water molecules in ice. When an antifreeze protein binds to an ice crystal, it doesn't melt the ice. Instead, it changes the shape of the crystal, making it harder for additional water molecules to join the growing lattice. This creates what's called thermal hysteresis, a gap between the melting point and the freezing point of the solution. Normally, water freezes and melts at the same temperature, 0 degrees Celsius. But in the presence of antifreeze proteins, water might not freeze until it reaches -5 degrees, even though it still melts at 0. This gives organisms a buffer, a range of temperatures, where they can remain active, even though ice is present. Antifreeze proteins from glacier microbes are particularly interesting because they're often hyperactive, meaning they produce a large amount of thermal hysteresis even at low concentrations. Some bacterial antifreeze proteins can lower the freezing point by several degrees Celsius, far more than most fish antifreeze proteins. This hyperactivity is thought to be due to the proteins binding not just to one face of an ice crystal, but to multiple faces, effectively coating the crystal and preventing it from growing in any direction. The exact mechanisms are still being studied, but it's clear that these molecules are exquisitely tuned to their environment. In addition to preventing ice from growing, antifreeze proteins also prevent ice recrystallization, the process by which small ice crystals merge into larger ones. Recrystallization is particularly damaging to cells because large ice crystals have sharp edges that can puncture cell membranes. By inhibiting recrystallization, antifreeze proteins help keep ice crystals small and relatively harmless. This is one reason why organisms that produce antifreeze proteins can survive being frozen solid. They don't prevent ice from forming entirely. They just control its growth and structure. Antifreeze proteins are just one part of the psychrophile toolkit. These organisms also produce cold shock proteins, molecules that help cells respond to sudden drops in temperature, and cold acclimation proteins, which are continuously produced during long-term growth in the cold. They're enzymes, the molecular machines that carry out chemical reactions in cells, have evolved to work efficiently at low temperatures. Enzymes from organisms that live in warm environments become sluggish and ineffective in the cold, but psychrophile enzymes remain active. This is partly because their three-dimensional structures are more flexible, allowing them to change shape and catalyze reactions, even when molecular motion is reduced by cold. The cell membranes of psychrophiles are also adapted. At low temperatures, membranes made of typical lipids become rigid and brittle, which can be fatal. But psychrophiles produce lipids with shorter carbon chains and more unsaturated bonds, which keep the membrane fluid and flexible even in the cold. Some also produce compatible solutes, small molecules that don't interfere with cellular processes, but that lower the freezing point of the cell's interior, much like salting. The Rhodes prevents ice from forming. All of these adaptations work together, allowing psychrophiles to not just survive, but thrive at temperatures that would immobilize or kill most other organisms. Scientists have catalogued dozens of bacterial genera living in glaciers, each with its own suite of adaptations. Polyromonas, Xanthynobacterium, Sphingomonas, and Methylobacterium are among the most common. These bacteria have been found in glaciers all over the world, from the Arctic to Antarctica to the Himalayas. Some are photoautotrophs, using sunlight to produce energy. Others are chemolithotrophs, deriving energy from the oxidation of inorganic compounds like iron, sulfur, or nitrogen. Still, others are heterotrophs, feeding on organic matter produced by other organisms. Together, they form complex food webs, cycling nutrients and energy through the glacier ecosystem. One of the most surprising discoveries about glacier microbes is that many of them remain active even when frozen. For a long time, scientists assumed that life essentially stopped below the freezing point, that microbes in frozen ice were in a state of suspended animation. But recent studies using advanced techniques have shown that this isn't true. In samples taken from glaciers in Iceland and Greenland, researchers found that more than 50% of bacterial cells were translationally active, meaning they were actively synthesizing proteins, even when the snow and ice they lived in was frozen. And when frozen samples were brought back to the lab and thawed, the bacteria could resume full activity within 24 hours. They weren't sleeping, they were just waiting. This finding has profound implications. It means that the biological and biogeochemical processes happening on glaciers are shaped not by long-term seasonal cycles, but by short-term, day-to-day, or even hour-to-hour fluctuations in temperature, light, and moisture. During a warm, sunny day, meltwater flows across the glacier surface, carrying nutrients and stirring up microbial activity. At night, when temperatures drop, the surface refreezes, but the microbes don't shut down entirely. They remain alert, ready to resume full activity as soon as conditions improve. This rapid responsiveness to changing conditions suggests that glacier ecosystems are far more dynamic and resilient than previously thought. It also means they're vulnerable to the changing melt regimes brought about by climate change. As glaciers experience more intense and prolonged melting periods, the microbial communities on their surfaces will change in ways we're only beginning to understand. Now, let's turn our attention to one of the most unusual and charismatic inhabitants of North American glaciers, the iceworm. Iceworms belong to the genus Mesenchytraeus and are relatives of earthworms, though they're much smaller. The most well-known species is Mesenchytraeus solifigus, which lives on coastal glaciers from south-central Alaska down through British Columbia, Washington, and Oregon. The species name, solifigus, means sun-avoiding, and it's an apt description. Iceworms spend most of their time hidden within the glacier, emerging on the surface only in the evening and early morning when the light is dim. Expose an iceworm to direct sunlight or warm temperatures, and it will quickly burrow back into the ice. Iceworms are small, usually less than a centimeter long, and as thin as a strand of dental floss. They're dark, almost black, which makes them easy to spot against the white ice, at least if you know what you're looking for and visit the glacier at the right time of day. Despite their diminutive size, iceworms can be astonishingly abundant. On Seattle Glacier in Washington state, population counts indicated over 7 billion iceworms. That's more than the entire human population of Earth, all living on a single glacier. On other glaciers, densities can reach several thousand worms per square meter. When you walk across the surface at dusk, the ice seems to writhe with them, tiny dark threads wriggling through the snow and meltwater. Ice worms have one of the narrowest temperature tolerances of any animal on Earth. They thrive at 0 degrees Celsius, right at the freezing point of water. Go a little warmer and they begin to struggle. At temperatures above 5 degrees Celsius, their bodies literally start to decompose. A whole process called autolysis, where the cells produce digestive enzymes and self-destruct. Go a little colder and they freeze solid, though they can survive being frozen down to about -7 degrees Celsius. This narrow window means that ice worms are restricted to temperate glaciers, where the ice temperature hovers near 0 year-round. They're not found in the colder, more continental glaciers of the interior mountain ranges, or the polar ice caps. Their range is limited to coastal regions, with mild winters and cool summers. How do ice worms move through the ice? Scientists aren't entirely sure. Some believe they travel through microscopic fissures and channels in the ice, squeezing between tightly packed ice crystals. Others think they secrete chemicals that slightly lower the freezing point of the ice around them, creating a thin film of liquid water they can move through. What's certain is that they use tiny bristles called setae, found on the outside of their bodies, to grip the ice and pull themselves along. They can move at a speed of about 3 meters per hour, which is slow by most standards, but impressive for a worm living in solid ice. Ice worms feed on snow, algae, bacteria, and organic detritus that accumulates on the glacier surface. They're an important part of the glacier food web, recycling nutrients and serving as prey for birds and other animals that visit the glacier. Their abundance suggests that they play a key role in nutrient cycling on the glacier, converting microbial biomass into forms that can be transported downstream or consumed by higher-level predators. But perhaps the most fascinating thing about ice worms is their metabolism. Most cold-blooded animals become sluggish as temperatures drop because their cellular processes slow down. Ice worms are the opposite. As temperatures decline towards 0, their intracellular ATP levels, the energy currency of the cell, actually increase. This is a paradox, a seeming violation of the normal rules of biochemistry. Exactly how ice worms achieve this is still being studied, but it likely involves unique adaptations in their mitochondria, the cellular structures that produce ATP, and in the enzymes that manage energy metabolism. Ice worms are packed with melanin, the same dark pigment found in human skin, and some researchers speculate that this melanin might help them harvest energy from sunlight, though this idea is still speculative. Ice worms are also genetically isolated, because they can't travel far from the glaciers they live on. Populations on different glaciers have little or no gene flow between them. Over time, this has led to the formation of distinct genetic clades, groups of related populations that are more closely related to each other than to worms on other glaciers. There are northern and southern clades, separated by geography and evolutionary history. And within these clades, each glacier has its own unique population. This isolation makes ice worms vulnerable to extinction. If a glacier melts entirely, the ice worms living on it have nowhere to go. They can't migrate to another glacier. They simply disappear. As climate change accelerates glacier melting, ice worms are in trouble. Glaciers that once supported thriving populations are shrinking or vanishing altogether. Researchers are racing to study these worms before they're gone, but the window of opportunity is closing. Ice worms are a reminder that glaciers aren't just frozen water. They're habitats, home to species found nowhere else on Earth. When scientists drill ice cores from glaciers, extracting long cylinders of ice that can be thousands of years old, they're looking for clues about past climates. Tiny bubbles of ancient air trapped in the ice can reveal what the atmosphere was like centuries or millennia ago. Layers of dust and ash mark volcanic eruptions and other events. But buried in that ice, frozen in time, is something else: microorganisms. Ice cores from glaciers around the world have revealed bacteria, fungi, and viruses that have been entombed for thousands, even tens of thousands of years. Some of these microbes are still alive, or at least viable, meaning they can be revived if the ice is melted and the cells are given the right conditions. Others are clearly dead, their cellular machinery broken down beyond repair, but their DNA remains intact, a genetic archive waiting to be read. In ice cores from the Tibetan Plateau, scientists found microorganisms dating back nearly 15,000 years. Analysis of these ancient microbes revealed bacterial genera like Xanthinobacterium, Polyromonas, Homaniomonas, Flavobacterium, Sphingomonas, and Methylobacterium, many of the same genera found in glaciers today. Perhaps even more intriguing, the ice cores also contained viruses, specifically bacteriophages, viruses that infect bacteria. These viral genomes were sequenced, and analysis revealed that they belonged almost entirely to groups of viruses never before seen by science. Of the 33 viral types identified, 28 represented novel genera. Not a single viral species was shared with any of the 225 environmental viromes, collections of viral genetic material that scientists had previously analyzed from diverse environments around the world. These viruses are unique to glacier ice, and many of them are what's called temperate phages, viruses that can integrate into their host's genome and lie dormant until conditions are favorable for reproduction. This is significant because it suggests that temperate phages are particularly well suited to life in glacier ice. In most environments, viruses that kill their hosts immediately are more successful. But in glacier ice, where opportunities for infection are limited and conditions are often harsh, a virus that can lie low and wait for better times may have an advantage. The presence of these viruses also tells us that the glacier isn't just a graveyard for ancient microbes. It's a functioning, if extremely slow, ecosystem with interactions between hosts and parasites, predators and prey, shaping the community over time. Some of the glacier viruses carry genes that suggest they can help their bacterial hosts. These are called auxiliary metabolic genes, and they code for proteins that can enhance the bacteria's ability to survive or compete. For example, some of the viruses carried genes involved in motility, the ability to move. This might help infected bacteria find nutrients or escape unfavorable conditions. Other viruses carried genes involved in nutrient acquisition or stress response. In a sense, these viruses are symbiotic partners, not just parasites. They contribute to the survival of their hosts, and in return, they get a safe place to replicate and spread. The discovery of ancient microbes and viruses in glacier ice has implications beyond glacier biology. It tells us that life can remain viable for extremely long periods in frozen conditions, which is relevant to the search for life on other planets. Mars, for example, has polar ice caps and subsurface ice deposits. If life ever existed on Mars, could it have been preserved in the ice, waiting to be discovered by future explorers? Europa and Enceladus, moons of Jupiter and Saturn, have liquid water oceans beneath thick shells of ice. Could microbial communities thrive in those alien seas, sustained by geothermal energy from the moon's interiors? By studying the microbes of Earth's glaciers, astrobiologists are learning what to look for and how to interpret the signs of life in icy environments elsewhere in the solar system. Perhaps the most extreme and least accessible habitats in glaciers are the subglacial lakes, bodies of liquid water that exist beneath hundreds or thousands of meters of ice. These lakes were first detected decades ago using radar and seismic surveys. But it wasn't until the 21st century that scientists developed the technology to drill down through the ice and sample them directly. Doing so is extraordinarily difficult. The drilling must be done cleanly, without introducing contaminants from the surface, which means using specially designed hot water drills that are sterilized and filtered. The water used for drilling is treated with ultraviolet light to kill any microbes it might contain, and the equipment is carefully cleaned to avoid contamination. Even with these precautions, there's always a risk that what you find in a subglacial lake isn't native to the lake, but was accidentally introduced during drilling. In 2013, a team of scientists successfully accessed subglacial Lake Whillans in West Antarctica, located beneath 800 meters of ice. The lake is only about 6 feet deep and covers roughly 20 square miles, kept liquid by heat from the bedrock below and friction from the glaciers moving over it. When the scientists analyzed water and sediment samples from the lake, they found a thriving microbial community, far more diverse than anyone had expected. The lake harbored nearly 4,000 distinct types of microorganisms, classified into operational taxonomic units based on their genetic material. The microbes in Lake Whillans are chemoautotrophs, meaning they get their energy not from sunlight, which never reaches the lake, but from chemical reactions involving minerals dissolved in the water. Specifically, they oxidize compounds containing nitrogen, iron, and sulfur, extracting energy from the transfer of electrons. This is similar to how some bacteria in deep-sea hydrothermal vents survive, and it demonstrates that life doesn't require sunlight to thrive. As long as there are chemical gradients to exploit, sources of energy in the form of reduced compounds, life can persist. The discovery of life in Lake Whillans was groundbreaking, but questions remained about whether the microbes were truly native to the lake or had been introduced during drilling. Those doubts were put to rest when scientists successfully sampled another subglacial lake, Lake Mercer, in 2018. Lake Mercer lies beneath over 1,000 meters of ice and has been sealed off from the surface for millennia. The drilling process was even more carefully controlled, and the results were even more astonishing. Over 1,300 single-cell genomes were sequenced from Lake Mercer, representing bacteria and archaea. Most of these genomes corresponded to new species, organisms that had never been identified before. Phylogenetic analysis, the construction of evolutionary trees based on genetic similarity, showed that the Lake Mercer microbes were genetically isolated from marine and surface biomes. They weren't recent arrivals, blown in by the wind or carried by meltwater. They were unique lineages that had been evolving in isolation beneath the ice for thousands of years. The microbes in Lake Mercer show a range of metabolic strategies. Some are heterotrophs, feeding on organic carbon that was deposited when the region was last connected to the ocean about 6,000 years ago. Analysis of the carbon in the sediments revealed the presence of carbon-14, a radioactive isotope that decays over time. The presence of carbon-14 means that the organic matter isn't ancient, at least not in geological terms. It's from the Holocene, the current geological epoch, and the microbes are still feeding on it, recycling nutrients that have been locked away beneath the ice for millennia. Other microbes in the lake are chemolithotrophs, oxidizing iron, sulfur, or nitrogen compounds. The lake's microbial community varies depending on oxygen concentration. In areas with more oxygen, aerobic organisms dominate. In areas with less oxygen, anaerobic organisms take over, using nitrate or iron as electron acceptors instead of oxygen. Subglacial lakes aren't entirely isolated from one another. Water flows between them through subglacial channels, creating a dynamic hydrological system. When one lake fills up, it can overflow into another, carrying microbes and nutrients with it. This connectivity means that subglacial microbial communities can spread and exchange genetic material over large distances, even though they're sealed off from the surface. It also means that the total number of microbes beneath the Antarctic ice sheet is staggering. Estimates suggest that the sediments beneath the ice sheet could contain somewhere between 10 to the 27th and 10 to the 28th cells, more than the number of microbes in all the world's freshwater ecosystems combined. The existence of these subglacial ecosystems raises profound questions. How long have they been isolated? How did they originally form? And how do they persist, given the extreme limitations on nutrients and energy? The answers are still being worked out, but it's clear that subglacial lakes are among the most extreme and least understood ecosystems on Earth. They're also among the most vulnerable. As climate change causes glaciers to retreat and melt, some subglacial lakes may drain or be exposed to the surface for the first time in thousands of years. When that happens, the unique microbial communities they harbour could be lost forever, or they could mix with surface communities, forever altering their evolutionary trajectory. As we've seen, life in glaciers employs a stunning array of survival strategies, each finely tuned to the specific challenges of the icy environment. But these strategies can be grouped into a few broad categories. First, there's freeze avoidance, the strategy of preventing ice from forming in the first place. This is what antifreeze proteins do. By binding to ice crystals and preventing them from growing, these proteins allow organisms to remain liquid even at sub-zero temperatures. Freeze avoidance works well as long as temperatures don't drop too far, but it has limits. If the temperature falls below a certain threshold, ice will form regardless, and the organism must either tolerate it or die. This brings us to the second strategy, freeze tolerance. Some organisms don't try to avoid freezing. Instead, they let ice form, but they control where and how it forms, so that it doesn't cause lethal damage. For example, some bacteria and algae can tolerate having ice crystals form in the spaces between cells, but not inside them. The ice outside the cells draws water out of the cells through osmosis, concentrating the cell's contents and lowering the intracellular freezing point. This dehydration is stressful, but it's survivable, and when the ice melts, the cells rehydrate and resume normal activity. Freeze tolerance is common among organisms that experience repeated freeze-thaw cycles, like those living on the surface of a glacier, where daily temperature swings can be dramatic. A third strategy is dormancy, when conditions are truly unfavorable, when temperatures are too low, nutrients too scarce, or water too limited. Many glacier organisms simply shut down. They stop growing, stop dividing, and enter a state of metabolic suspension. In this dormant state, they can survive for months, years, or even millennia, waiting for conditions to improve. Dormancy is particularly common among bacteria, many of which can form spores, highly resistant resting cells, that can endure extremes of temperature, radiation, and desiccation. But even non-spore-forming organisms can enter dormancy, reducing their metabolic activity to a bare minimum and waiting out the hard times. Nutrient scarcity is another major challenge for glacier organisms. Glaciers are oligotrophic environments, meaning they're extremely low in nutrients like nitrogen, phosphorus, and organic carbon. To cope, glacier microbes have evolved highly efficient nutrient uptake systems, capable of scavenging trace amounts of nutrients from their surroundings. They also recycle nutrients internally, breaking down and reusing cellular components when external sources are unavailable. Some glacier bacteria are chemolithotrophs, capable of deriving energy and building blocks from inorganic compounds like ammonia, hydrogen sulfide, or ferrous iron. This allows them to bypass the need for organic carbon entirely, making them independent of photosynthesis and able to thrive in total darkness. Light limitation is another challenge, particularly for organisms living deep in the ice or beneath a glacier. Photosynthesis is impossible in these environments, so organisms must rely on chemosynthesis, the production of organic matter from inorganic compounds using chemical energy. Chemosynthetic bacteria play a key role in glacier ecosystems, serving as primary producers in the absence of light. They oxidize reduced compounds delivered by meltwater or released from the weathering of bedrock, and they use the energy from these reactions to fix carbon dioxide into organic molecules. This organic matter then supports a food web of heterotrophic bacteria, fungi, and even small animals. Temperature adaptation goes beyond just preventing ice damage. Psychrophiles must also ensure that their enzymes remain active at low temperatures. Enzymes are proteins that catalyze biochemical reactions, and they work best at a specific temperature, their optimal temperature. For organisms living in warm environments, that optimal temperature is high, often above 30 degrees Celsius. But for psychrophiles, the optimal temperature is near or below zero. This is achieved through subtle changes in the enzyme's three-dimensional structure, making it more flexible and allowing it to change shape more easily, even when molecular motion is slow. Psychrophile enzymes also tend to have lower activation energies, meaning they require less energy to catalyze a reaction, which is crucial when thermal energy is limited. Cell membrane fluidity is another critical adaptation. At low temperatures, the lipid bilayers that make up cell membranes can become rigid and brittle, impairing their function. Psychrophiles counteract this by producing lipids with shorter carbon chains and more unsaturated bonds, both of which lower the membrane's transition temperature, the temperature at which it shifts from a fluid to a solid state. Some psychrophiles also produce carotenoid pigments, which insert into the membrane and help maintain its fluidity. Interestingly, these same pigments, like astaxanthin, also serve as UV protectors, demonstrating how a single adaptation can serve multiple functions. Finally, there's the question of how glacier organisms protect their genetic material. DNA is vulnerable to damage from radiation, oxidative stress, and freezing, all of which are common in glacier environments. To cope, glacier microbes have robust DNA repair systems, capable of fixing breaks, mutations, and other damage. Some also produce protective compounds that stabilize DNA and proteins, preventing them from degrading even during long periods of dormancy. The fact that viable microbes and intact viral genomes can be recovered from ice cores thousands of years old is a testament to the effectiveness of these protective mechanisms. As the world warms, glaciers are melting at an accelerating pace. In the Arctic, in the Alps, in the Andes, and in Antarctica, ice is disappearing. The implications for human societies, for water resources, for sea level rise are profound and widely discussed. But less often talked about is what glacier melting means for the unique ecosystems that exist within the ice. As glaciers retreat, the microbial communities they harbour are being lost. Some species may be able to migrate to other glaciers, carried by wind or water. But many, particularly those in isolated or rapidly disappearing glaciers, will simply vanish. This represents a loss of biodiversity, a loss of genetic resources, and a loss of knowledge about how life adapts to extreme conditions. Recognizing this, scientists are working urgently to document glacier microbial communities before they're gone. This effort involves collecting samples from glaciers around the world, sequencing the genomes of the organisms found there, and preserving samples in biobanks, refrigerated repositories where microbial cultures and DNA can be stored for future study. The goal is to create a library of glacier microbes, a snapshot of this unique biome before it disappears. This work is painstaking and expensive, requiring expeditions to remote, dangerous locations and sophisticated laboratory analysis. But it's essential. Once a glacier melts, the organisms that lived within it are gone forever, and with them go the insights they might have offered into evolution, adaptation, and the limits of life. There's also a more practical reason to study glacier microbes. Many of them produce compounds with potential commercial applications. Antifreeze proteins, for example, are already being used in the frozen food industry to prevent ice crystal formation and improve texture. They also have potential applications in organ transplantation, where keeping organs cold but not frozen is critical, and in cryopreservation, the long-term storage of cells and tissues at ultra-low temperatures. Enzymes from psychrophiles, which remain active at low temperatures, are useful in biotechnology, particularly in processes that need to be carried out in the cold to prevent degradation of sensitive compounds. And the pigments produced by snow algae, like astaxanthin, are valuable as antioxidants and nutritional supplements. Beyond the practical applications, glacier microbes are also of interest to astrobiologists, scientists who study the possibility of life on other planets. As mentioned earlier, icy moons like Europa and Enceladus may harbour liquid water oceans beneath their frozen surfaces. If life exists in those oceans, it's likely to be microbial, and it's likely to face challenges similar to those faced by glacier microbes on Earth. By studying how life survives in Earth's glaciers, astrobiologists can develop strategies for detecting life in extraterrestrial ice. They can also build instruments and design missions that are better suited to finding and characterizing alien microbes. The melting of glaciers also raises ethical questions. Do we have a responsibility to preserve glacier ecosystems, or at least to document them before they disappear? Should we be trying to save glacier-dependent species, like ice worms, perhaps by translocating them to more stable glaciers or keeping them in captivity? Or is it inevitable that as the climate changes, some species will be lost, and we should focus our efforts on mitigating the broader impacts of climate change rather than trying to save every individual ecosystem? These are difficult questions with no easy answers, but they're worth asking. What's clear is that glaciers are not lifeless. They're ecosystems, complex and dynamic, supporting a remarkable diversity of organisms that have evolved ingenious solutions to some of the most extreme conditions on Earth. From the cryoconite holes on the surface, where cyanobacteria and algae thrive in miniature pools of meltwater, to the subglacial lakes far beneath the ice, where bacteria feed on minerals in perpetual darkness. Glacier life is everywhere. It's in the red snow that colours alpine meadows in summer. It's in the ice worms that emerge at dusk to graze on algae. It's in the ancient microbes frozen in ice cores, waiting to tell their stories. And it's in the viruses, invisible but omnipresent, shaping the evolution and ecology of their bacterial hosts. The resilience of glacier life is humbling. These organisms have persisted through ice ages and interglacials, through volcanic eruptions and meteor impacts, through every environmental upheaval Earth has thrown at them. They've adapted to temperatures that would kill most other life forms, to darkness that lasts for months, to starvation rations that would leave most organisms unable to reproduce. And yet, they endure, and in many cases, they thrive. They remind us that life is not fragile. It's remarkably tough, capable of finding footholds in the most unlikely places and exploiting opportunities that would seem too meager to sustain anything. But glacier life is also vulnerable. It depends on the existence of ice, and as that ice melts, its habitat disappears. For some glacier species, particularly those with limited dispersal ability or narrow environmental tolerances, the future is uncertain. They may not be able to adapt quickly enough to keep pace with the changing climate, and they may not be able to find suitable habitat elsewhere. The loss of these species would diminish the richness and complexity of Earth's biosphere, and it would close off avenues of scientific inquiry that we've only just begun to explore. So, as you drift off to sleep tonight, think about the glaciers. Picture those vast expanses of ice and snow, gleaming under the midnight sun or shrouded in polar darkness. Know that beneath the surface, within the ice, and at the very bottom where the glacier meets bedrock, life is happening. Microbes are photosynthesizing, feeding, reproducing, and dying. Ice worms are wriggling through the snow. Viruses are infecting bacteria. Nutrients are cycling. Ecosystems are functioning, just as they have for thousands of years. And think about what it means that life can survive, even flourish, in such an extreme and inhospitable place. It means that life is adaptable, resilient, and creative. It means that wherever there's energy to harvest and resources to exploit, life will find a way. And it means that our planet, even its coldest, most barren-looking corners, is teeming with wonders waiting to be discovered. If you enjoyed this journey into the icy depths and learned something new about the hidden ecosystems of glaciers, please consider giving this video a like and subscribing to the channel. Your support helps us continue exploring the quiet wonders of the natural world. And if you have suggestions for future topics, or if there's a particular aspect of biology that fascinates you, leave a comment below. We love hearing from you. Until next time, rest well, and may your dreams be filled with the serene beauty of snow-covered peaks and the quiet marvels of life in the ice.